Good afternoon, and welcome to this edition of AFTD's Healthcare Professional Educational Webinar Series, Approaching a Cure, FTD Genetics and Clinical Trials. I'm Dr. Shana Dodge, AFTD's Director of Research Engagement. On behalf of AFTD, I thank you all for joining us. Please note that our audience will be muted throughout this webinar to keep background noise to a minimum. If you're experiencing any technical issues, please write a message in the questions box, and a member of the AFTD staff will try to address them. You can also use the questions box to submit questions for our presenters. There will be a brief Q&A portion of this webinar where they will answer as many questions as they can. Feel free to submit your question as it occurs to you. You don't have to wait until the Q&A portion to ask your question. Additionally, this webinar will be recorded and archived for later viewing on AFTD's YouTube page. Before we get started, I have a few quick announcements. First, AFTD is hard at work planning our 2024 education conference. Registration won't open until the new year, so for now, you can save the date. The conference will be held Friday, May 3rd in Houston. This annual conference brings together persons affected by FTD, experts in the field, and other healthcare professionals for a full day of learning and connection. If you can't make it to Houston, you have the option of following along with our free live stream. Additionally, most sessions will be recorded for later viewing. Next, I'd like to highlight an AFTD resource for healthcare professionals called Partners in FTD Care. Each issue takes a deep dive into a particular disorder or specific aspects of care. Recent issues have focused on transitioning to residential care, the African American FTD experience, and what families should know and do after getting a diagnosis. There are over 30 issues of Partners in Care available by accessing the web address at the bottom of your screen. For healthcare professionals watching today's webinar live, we're offering continuing education credits and continuing medical educational credits via Rush University. This is part of AFTD's efforts to educate healthcare professionals on FTD and FTD care. Tomorrow, you will receive an email explaining how to apply for your CEC or CME. Please note you have 10 days from the date of the activity to complete the evaluation and claim credit. At the conclusion of this webinar, you should be able to identify 3 primary genes underlying FTD and why healthcare providers should consider genetic counseling for their patients with FTD, describe the current clinical trial landscape for genetic FTD, and explain how active clinical trials can inform future scientific breakthroughs across the FTD spectrum. Additionally, there are materials related to this program located in the handouts box on your screen. You will also get an email next week containing links to these materials. Now I'd like to introduce today's presenters. Dr. Peter Ljubenkov is a behavioral neurologist and clinical trialist at the UCSF Memory and Aging Center, where he serves as an associate program director of the Behavioral Neurology Training Program Fellowship. He has research interests in fluid biomarker and therapeutic drug development in persons with Alzheimer's disease and FTD, and has served as primary investigator for multiple interventional trials within these cohorts. Kim Jenny is AFTD's Manager of Genetic Initiatives. She has more than 20 years of experience as a genetic counselor working in a variety of settings, including pediatric genetics, commercial genetic laboratory services, and clinical consultation. As the Manager of Genetic Initiatives, Kim helps develop and implement AFTD initiatives related to genetic forms of FTD. And now I will turn things over to Kim.
Thank you, Shana, and welcome to everyone. Here are my disclosures. Next slide, please. So we all know that there are many forms of dementia, but FTD is unique in several ways, which make it difficult to diagnose, resulting in FTD being underdiagnosed, perhaps significantly so. First, FTD has a younger age of onset. It's the most common form of dementia in people under the age of 60. Symptoms can begin at any time in life, but most people develop symptoms in their late 40s to early 60s, a time that's critical when people are most engaged with their family, their careers, and finances. Next, the symptoms of FTD are different. Typically, they involve changes to behavior, language, or movement, but memory might not be affected. Third, FTD is considered somewhat rare, with about 50 to 60,000 people diagnosed in the US. However, experts believe the incidence is likely much higher. Again, FTD is underdiagnosed. There are currently no approved treatments available, so why is it important for families to know the exact diagnosis? Well, now more than ever, a correct and prompt diagnosis is critical to ensure that families can access not only care and resources, but potentially experimental treatments that are being developed. Finally, there's a stronger genetic contribution to FTD than in other forms of dementia, and many of the genes that cause FTD are dominantly inherited with a high degree of penetrance, meaning those with a pathogenic variant are likely to develop FTD, and their family members also have a 50% chance of developing FTD. Treatments that are currently being developed focus primarily on genetic forms of FTD, meaning those with a genetic cause for their FTD have the greatest chance to qualify for experimental treatments, and their participation in research is our greatest hope to develop effective treatments. Next slide, please. I mentioned the genetic contribution to FTD is stronger than in other forms of dementia, but not all FTD is inherited. We basically think of FTD as falling into three different categories. The majority, perhaps about 60% of FTD, appears to be sporadic. These are families with only one person diagnosed. No one else in the family has FTD or a related condition. And the presumption is that the relatives are not at risk. However, in a portion of people with an apparently sporadic form of FTD, these are people with no family history, we can still identify a genetic cause. Sometimes this is because the family history is limited or incomplete, or perhaps someone was misdiagnosed in the past. In a small portion of people, it may be due to a de novo genetic variant. In these individuals, the genetic variant that causes FTD occurred for the first time in them. Their genetic form of FTD may not be inherited, but importantly, it can be passed on to the next generation. Thus, a genetic evaluation is valuable even for those with an apparently sporadic form of FTD. About 40% of people diagnosed do have a family history that's suggestive of a genetic cause. It's always important to ask if there are other relatives diagnosed with FTD or related conditions like ALS, which is genetically linked to FTD. Perhaps someone has a mental health diagnosis like bipolar or depression, or there may be a relative who's having problems either understanding or producing language. Or someone in the family could have a diagnosis of a movement disorder like Parkinson's disease or some other form of dementia. When we see FTD and these related conditions clustering together in a family, we refer to this as familial FTD, and we're much more likely to identify a genetic cause in these families. When the genetic cause is identified, we can confidently refer to this as genetic FTD. An FTD-causing genetic variant can be found in about 20% of people with FTD. If there is a strong family history of FTD, but genetic testing is negative, it's most likely because there are additional genes that cause FTD that have not yet been identified. In these families, DNA banking might be an option to consider, and I'll mention that again in a bit. Next slide. There are many genes that can cause FTD, and there are more to be discovered. The three most common genes include chromosome 9, open reading frame 72, or C9orf72, the microtubule-associated protein tau, or MAPT, and progranulin, sometimes called GRN. C9orf72 is believed to be the most common genetic cause of FTD and genetic ALS. Most of these genes can present with any of the symptoms associated with FTD. This means that in one family, you may be seeing a person with behavioral changes, but there may be other relatives with either language or movement problems, again, contributing to the difficulty in diagnosing FTD. Genetic FTD follows a dominant pattern of inheritance. This means that both men and women are equally likely to develop signs and symptoms of FTD, and they have an equal chance of passing FTD on to their children. And most of these genes are highly penetrant. Therefore, a person with a pathogenic variant is likely to develop FTD. Each child of a person with genetic FTD has a 50% or a 1 in 2 chance to inherit the genetic variant that causes FTD. So clearly, a person's genetic status has important implications for the extended family. Next slide, please. Genetic counseling is always recommended before deciding about testing. There's never an expectation that a person must test. Genetic counselors focus on the entire family, and we bring a unique skill set that complements and enhances the care other health professionals provide. We're trained to educate and convey the nuances and complexities of FTD and genetic information to patients, their families, and even other healthcare professionals. Genetic counselors can also help unaffected relatives plan for the future and understand genetic privacy laws, including what is and is not protected under the Genetic Information Non-Discrimination Act, known as GINA. There's no right or wrong decision about testing, and if someone does want to test, there are different options available. A genetic counselor can help determine which test is the most robust and appropriate for a person. Importantly, if we do not know the results for the affected person, the person who has symptoms of FTD, then testing their unaffected relatives is not reliable. Again, DNA banking is a relatively easy and affordable option for when a person with FTD is either reluctant or unable to test. And a genetic counselor can help facilitate DNA banking. Often, genetic test results are difficult to interpret. Genetic counselors are adept at deciphering the significance of a person's results. And if a test result opens the opportunity to participate in research, a genetic counselor can help facilitate a person's participation in an appropriate experimental protocol. Next slide, please. So if the clinical picture and the family history all make sense, is there an added value to having that genetic test result? Well, first, FTD is difficult to diagnose, and a positive genetic test result confirms that the diagnosis is correct. Understanding one's genetic risk can inform numerous important life decisions, including family and financial planning, and career and lifestyle choices. And we know that the anxiety of not knowing if FTD is genetic can weigh very heavily not only on the person, but also their family. A known genetic risk can expedite a complicated diagnostic process and lead to quicker access to services and care. And it can help a person have more control around issues like how much longer can I work? And is it still safe for me to be driving? Knowing the genetic cause for FTD allows for predictive testing for relatives who may be at risk. And finally, identifying the specific genetic cause may open up opportunities to access some of those experimental treatments. And finally, those with genetic FTD are likely to be first in line when new treatments are approved by the FDA. Next slide. So, providers can help families prepare for a genetic evaluation by referring them to the AFTD website, where we have a wealth of information and resources about genetic counseling, testing, and research. Genetic counseling offers people the tools needed to better cope with the emotional and psychological aspects of living at risk for or with FTD. And AFTD is here to share resources and support for those impacted by FTD. Our community has shared that they are hungry for information about their genetic status. Information is power, and understanding the genetic basis for familial FTD empowers people to better manage their FTD journey in what is often a very chaotic experience. The overwhelming majority of people living with FTD and their care partners have expressed interest in understanding their genetic status. Research indicates that people are resilient even when faced with a positive genetic test result. Overall, when a person knows their status, their anxiety may actually decrease compared to the degree of anxiety they felt before their status was known. There's never been more hope for a future where FTD can be treated or even cured. New drugs have recently been approved for ALS and Alzheimer's, but more clinical trials are needed to develop treatments for FTD, and we need participants for those clinical trials for them to be successful. AFTD and the FTD Disorders Registry are reliable sources of information about what trials are currently available, and we will stay abreast as these opportunities change. Researchers are developing treatments that target specific FTD-causing genes, and people with variants in those genes are uniquely eligible for some of the clinical trials currently underway. Once a gene-specific treatment is successful, we may have a better understanding for how to develop treatments for all forms of FTD, both genetic and apparently sporadic. In summary, when a person is given the opportunity to learn their genetic status, there's the potential to benefit the individual, the family, and future generations. Thank you. And now it's my pleasure to turn things over to Dr. Ljubenkov.
Great, thank you so much. Let's see, I'm just going to share my slides here. Okay, can you all see my slides okay? Okay, great. Okay, hello everyone. Again, I'm Dr. Peter Ljubenkov. I'm a clinical trialist at UCSF Memory and Aging Center, and I'm going to be talking over the current landscape of clinical trial development in frontotemporal dementia with a special emphasis on clinical trials devoted towards genetic forms of frontotemporal dementia. Here are my disclosures. Just want to mention I do happen to be an investigator in some of the trials that I'm going to discuss today. And I want to start by saying that, in my opinion, we live in really exciting times for frontotemporal dementia therapeutic development. We don't have a disease-modifying therapy yet, but there are really good reasons to be excited. There's a large and growing infrastructure of international researchers, advocacy groups, federal programs, foundations, and industry partnerships that are all working to find a cure. And there are actually many exciting potential therapies that are in various stages of development right now. A lot to look forward to. I also want to mention there's a lot of good ways to stay in the loop about what I'm going to be talking about today. Of course, AFTD is always a great resource for basic education about frontotemporal dementia, as well as information about upcoming research. The FTD Disorders Registry is a great resource for patients and caregivers who are interested in staying in the loop about potential research projects that they might qualify for. And of course, clinicaltrials.gov is a great resource to look into trials for a particular diagnosis, including FTD variants. And I want to also start by saying that, highlighting the fact that we now live in a world in which there are multiple really large and dedicated multi-site consortia that are dedicated towards facilitating frontotemporal dementia clinical trials. In North America, we have All FTD. This is an NIH-funded project that joins together over 20 different sites of FTD research to enroll patients in longitudinal research for annual clinical visits, including imaging, data collection, and biospecimen collection. And really, the overall goal of this consortium is to enable the basic infrastructure in which to launch the next wave of frontotemporal dementia clinical trials, to build the expertise in these diseases and to build that infrastructure in which to mount these clinical trials. There's a similar consortium in Canada, in Europe and parts of Eastern Canada called GenFi, and there's another similar consortium that's more recent called REDLAT in Latin America. And the good news is that these consortia and a lot of other similar efforts really across the globe have a wonderful collaborative spirit and have joined forces in a larger initiative called the Frontotemporal Dementia Prevention Initiative, which is really intended to improve the inclusiveness of FTD research, enlarge the catchment area for research in rare familial forms of frontotemporal dementia, and really provide an international infrastructure in which to launch the next wave of frontotemporal dementia clinical trials and really bring us closer to finding that cure. Now, I'm going to have to get a little into the weeds about the complexity of FTD. Every therapeutics talk about FTD involves an obligatory slide like this, and Kim talked about some of these topics a little bit, but I want you to all to draw a distinction between clinical syndromes and pathological findings on autopsy. Frontotemporal dementia is a clinical syndrome, and there are 3 canonical variants of frontotemporal dementia. These are a set of clinical syndromes that a patient might experience. There's the behavioral variant of frontotemporal dementia, which starts with behavior changes first. There's the non-fluent variant of primary progressive aphasia, and there's a semantic variant of primary progressive aphasia, which involves conceptual loss. And these different forms of frontotemporal dementia can have a variety of different autopsy findings. And the primary autopsy finding associated with FTD is frontotemporal lobar degeneration. there's two main kinds of frontotemporal lobar degeneration. There's FTLD with tau, which involves accumulation of tau proteins in brain cells, and there's FTLD-TDP, which involves mislocalization of another protein called TDP43. There are also other rarer forms of frontotemporal lobar degeneration. Of these, something called FTLD-FUS is the most common. And of course, occasionally Alzheimer's disease can mimic the syndromes that we call frontotemporal dementia. Now, each of these clinical syndromes may have some association, relative predilection for some of these pathological findings on autopsy. The behavioral variant of frontotemporal dementia can be pretty diverse, whereas the non-fluent variant of primary progressive aphasia tends to be more closely associated with tau, and there's a very strong relationship between the semantic variant of primary progressive aphasia and TDP43 pathology. There are also other clinical syndromes that fit into the larger spectrum of frontotemporal lobar degeneration associated clinical syndromes. Of these, frontotemporal dementia with ALS features, so frontotemporal dementia motor neuron disease, strongly associated with TDP43 or FUS pathology, whereas cortical basal syndrome and progressive supranuclear palsy, these syndromes which may involve a host of symptoms, including movement changes, those syndromes tend to be more closely associated with tau pathology on autopsy. And then, as was mentioned by Kim, there are a variety of different genetic causes of frontotemporal dementia, including autosomal causes that have an autosomal dominant pattern of inheritance. And there are the three most common mutations are mutations in the MAPT gene, the tau gene itself, expansion of C9 open reading frame 72, and mutations in the progranulin gene. Tau mutations are associated with tau pathology, whereas C9 and progranulin are strongly associated with TDP43 pathology. And when you think about that complexity that I just showed in that last slide, something that might occur to you is that that complexity might present a sort of challenge in clinical trial development in frontotemporal dementia. This is because while we're very good at detecting Alzheimer's disease now prior to autopsy with PET scans and CSF and now emerging blood tests, we don't yet have robust and validated tools to distinguish between tau pathology and TDP43 pathology prior to autopsy. And for this reason, the clinical trial space to date has largely been focused on clinical syndromes that reliably predict a specific pathology on autopsy. So a lot of clinical trials to date have occurred in this disorder called progressive supranuclear palsy because it reliably predicts tau pathology in autopsy. There's now growing efforts to conduct more clinical trials in patients with semantic variant primary progressive aphasia, and that syndrome is strongly predictive of TDP43 pathology. And really, the familial FTD space is associated with a strong relationship with specific types of FTLD on autopsy. So C9 expansion and progranulin deficiency, they reliably predict TDP43 pathology on autopsy, whereas MAPT mutations reliably predict tau pathology in autopsy. And so this is so the familial FTD space has been a region of active development within FTD clinical trials. So I just want to start by talking about therapeutic development within TDP43 disorders. So patients with FTLD TDP experience this phenomenon where this protein called TDP43, a DNA binding protein that is important in splice regulation, is depleted in the nucleus of brain cells, and then also accumulates in the cytoplasm of brain cells. And when this occurs, the neurons become injured and die. And there's a variety of reasons why this might happen. We haven't quite connected all the dots in terms of why this occurs in familial forms of frontotemporal dementia. One of the many reasons that TDP43 might get mislocalized is something called oxidative stress, which is a type of chemical stress that can be released by a variety of processes. One of them is an overactive immune system, and this is one potential target for future therapies. For instance, there's one drug called verdiperstat, which targets a specific pathway by which oxidative stress might be produced by an overactive immune system by the microglial cells that are around brain cells in TDP43 disease. And this drug is actually being investigated in a trial that I'm currently leading, the Verity trial, This is a phase 1 trial in patients with semantic variant primary progressive aphasia. It's really the first clinical trial to specifically focus on patients with SVPPA and really rationalizes that cohort as a cohort that is strongly predictive of TDP-43 pathology. It's also the first clinical trial to really try to use the All-FTD Research Consortium that I mentioned earlier as a referral network to identify patients and route them to one of 5 participating sites, which I have highlighted in red. If you're a patient with SVPPA or you know somebody with SVPPA, please contact us. We'd be interested in talking to you. We're currently enrolling patients in our trial. So that was a trial in SVPPA. Now I want to talk about familial forms of TDP-43 pathology. And one of the regions of most active development for clinical trials in FTLD TDP is within the space of progranulin deficiency. These patients with progranulin deficiency are individuals with heterozygous progranulin gene mutations. As mentioned earlier, this is a highly penetrant cause of TDP-43 pathology, and the mechanism is haploinsufficiency. So patients who have pathogenic mutations are, because they have this mutation, their cells are making less progranulin, and their cells inside of their brain, including neurons and immune cells and supportive cells in the brain, are releasing less progranulin, of this protein called progranulin, into the extracellular space. And so the main therapeutic strategy that has been focused on has been a strategy to boost the available progranulin. And for this reason, cerebral spinal fluid tests for the progranulin protein have emerged as a rational biomarker for drug target engagement. So if you have a candidate drug and you think it might work in patients with progranulin deficiency, in that clinical trial, you're seeking to try to boost the detectable amount of progranulin in the cerebral spinal fluid. And there are really many drugs that are currently being investigated that could possibly boost progranulin in patients with progranulin deficiency. 4 of these drugs are in trial right now. 1 of them is about to enter into trial. And as I said, all of them are capitalizing on this main central strategy. The drug program that's probably furthest along in development is a drug called AL001. This is an antibody to something called sortilin. Sortilin is a protein that clears progranulin from the extracellular space. So by blocking sortilin, that can essentially make more progranulin available. And for what it's worth, in the phase 1 and 2 trials, patients with progranulin deficiency really did seem to experience a robust boost in their cerebral spinal fluid progranulin levels after getting this drug. So this is a figure from the phase 2 trial. This is not peer-reviewed data, by the way. This is data that was made publicly available by the trial sponsor. Again, this is not peer-reviewed data. This is data made available by the trial sponsor, and this is open label data, so you have to take it with a grain of salt. But for what it's worth, the individuals who were enrolled in that phase 2 trial were also compared to a historic control group of individuals with progranulin deficiency from the GenFi cohort, which I mentioned earlier. And for what it's worth, individuals enrolled in the phase 2 trial did seem to have a better clinical trajectory. So importantly, this drug is now being investigated in an active phase 3 trial that enrolls individuals with symptomatic progranulin deficiency and individuals with asymptomatic, individuals who are asymptomatic mutation carriers who have biomarker evidence that may suggest they're about to develop symptoms. And so that's one mechanism to potentially boost progranulin. Another mechanism is through gene therapy. And when I say gene therapy, the drugs that I'm talking about that are currently in clinical trials at the moment all capitalize on this one main mechanism, this adeno-associated virus vector system. So basically, to simplify things, you have the outer covering of a virus with the genetic material removed, and essentially the progranulin gene inside of that vector, and that vector is introduced into the CNS, usually through an intercisterna magna injection, so an injection to the cerebral spinal fluid at the base of the skull, and that gene therapy enters into the CNS and teaches a subpopulation of cells in the CNS to make more progranulin. And this is data, this isn't human data, this is data from non-human primates, but as you can see, multiple different AAV vectors do seem to be able to deliver this type of therapy and boost the expression of progranulin in the CNS. So there are actually 3 different drugs in this class that are either in trial already or about to enter into trial. The 2 first trials, I believe they've already completed enrollment, and I think the last on the list is about to start enrolling in Poland and Spain. So one other question that comes up is, is it possible to just give people progranulin, to literally infuse it through their blood? You can't really just infuse progranulin itself into the blood because because you have a blood-brain barrier, and it won't be able to get into the brain in sufficient quantities to have the intended effect. But there are potentially ways around this issue through something called the human transferrin receptor system. This is a means by which your body can actively transport target proteins from the blood into the central nervous system. And it turns out you can actually make a hybrid protein that involves progranulin, which is pictured in green, fused to another protein that interacts with that receptor. And that might be a mechanism to effectively deliver progranulin from a peripheral infusion into the brain. And there's actually a drug trial like that currently underway. So patients, this drug program has already completed its phase 1 component in healthy controls and is now about to be investigated in symptomatic individuals who carry progranulin gene mutations. So that's the clinical space for progranulin deficiency. I want to move on to C9 open reading frame 72 expansion. As was mentioned earlier, this mutation is the single most common autosomal dominant cause of familial ALS and FTD. We don't exactly know why, there's hypotheses, but we don't exactly know why this gene expansion gives rise to TDP43 pathology, but there are several potential mechanisms for cell toxicity. I do want to mention that when we say expansion, we literally mean that gene has become longer. So there's this hexanucleotide region that is repeated in our DNA. Typically, we have 11 or less repeats, but in somebody with pathogenic expansion, there could be hundreds of repeats. And that expanded region can give rise to RNA molecules that have that expanded region in them, and those expanded RNA molecules can themselves induce toxicity through certain mechanisms. And the interesting thing is that those expanded RNA molecules can actually be transcribed into these unusual proteins, these dipeptide repeat proteins, which themselves are thought to be able to exert some toxicity on brain cells. And actually, these dipeptide repeat proteins can be measured in the cerebral spinal fluid and have become another rational biomarker of drug target engagement. So in progranulin deficiency, I talked about trying to boost progranulin levels in the cerebral spinal fluid. In some C9 open reading frame 72 trials, the goal is for the drug to suppress the amount of detectable dipeptide repeat proteins in the cerebral spinal fluid. And so one of the trials to do that was a trial of a drug called AIT-101. This is essentially a proprietary form of another drug called apilimod, which is a molecule that was repurposed from oncology trials. It's something called a PIK5 inhibitor and probably has a variety of effects on cells. One of the effects is that it impacts something called autophagy, which is a mechanism by which cells can clear away broken down proteins. And so this trial was investigated in patients with C9 open reading frame 72 expansion and ALS, not frontotemporal dementia. And again, this is not peer-reviewed data. This was publicly presented by the trial sponsors, but reportedly the drug was safe and well tolerated in that cohort. And intriguingly, the drug seemed to robustly decrease the detectable dipeptide repeat proteins in the cerebral spinal fluid of individuals enrolled in that trial by about 73% on average. So this result is, the result of this trial has caused a lot of discussion in my field about the possibility of this drug being used in future FTD clinical trials, particularly in patients with C9 open reading frame 72 expansion. But because of its mechanism, it may have a role outside of C9 expansion because it has a role in autophagy and other cellular processes. And it actually, apilimod itself, the molecule apilimod, has been investigated in basic preclinical models of tauopathies as well. So it may have a role outside of just TDP-43 disorders. Other clinical trials that are, so there have been quite a few clinical trials in C9, and I don't have time to cover them. I'm just going to talk about the few that are still ongoing. Metformin, there's an ongoing trial of metformin, a diabetes drug. I believe this trial is completed enrollment. I'm still awaiting the readout of that, but in preclinical models, this drug was rationalized as yet another way to lower dipeptide repeat proteins. And then there's a whole other different mechanism that's being investigated in trials right now with a drug called TPN-101. So it's kind of an interesting drug. So, you probably all know we have DNA that is part of our genetic code, and DNA is used as a template for RNA molecules, and then RNA is used as a template for making proteins. But there are ways for RNA to become a template for DNA using something called reverse transcriptase. And we actually have these little coding regions in our own genome called retrotransposable elements that can jump back and forth between DNA and RNA, and those regions that use an RNA template can actually reinsert back into our DNA. And some of these retrotransposable elements can actually become dysregulated in some models of neurodegenerative disease, and they may actually be a mechanism of downstream neurotoxicity. It may not just be downstream of TDP-43 pathology, it may also be downstream of tau pathology. So this drug, TPN-101, is actually being investigated in 2 clinical trials that are about to wrap up very soon. So a phase 2A trial in patients with either ALS or FTD due to C9 open reading frame 72 expansion, and another phase 2A trial in patients with progressive supranuclear palsy. Again, that is a disorder that is strongly associated with tau pathology and autopsy. And so with that, I'm just going to finish up by talking about talking a little bit about clinical trial development in the FTLD tau space. As I alluded to earlier, most clinical trial development within frontotemporal lobar degeneration, tau, has occurred within this disorder, progressive supranuclear palsy. And as I said earlier, this is because this disorder is highly specific for tau pathology and autopsy. It's really been the focus of multiple clinical trials, including several large trials that are approaching the size of a pivotal trial. And actually, it's going to be the focus of an upcoming trial that hasn't started enrolling yet, testing a drug called Relyvrio. Relyvrio was just recently approved, by the way, for ALS. It's hypothesized to have a variety of neuroprotective roles through its role in impacting endoplasmic reticulum and mitochondrial function. And that's going to be the focus of an upcoming phase 2 trial, probably very soon. Unfortunately, there just haven't been a lot of clinical trials for MAPT mutation carriers. Some patients with pathogenic mutations have entered in small numbers into previous small trials, but there hasn't been a dedicated MAPT mutation carrier trial. And part of the reason for this is the relative rarity of this condition. I'm going to get into that in a few slides. And I want to say it's not for lack of interesting drug candidates. As many of you might know, Alzheimer's disease also involves a lot of tau pathology. And so there are a lot of therapeutic modalities that are currently being investigated by multiple different groups. Some of them are, most of them are being investigated in Alzheimer's disease, some of them in PSP as well, but there are a lot of different potential ways to mitigate tau pathology. And so there are a lot of different things that could be tried in clinical trials in MAPT mutation carriers. I would say one of the most interesting drugs, at least in my opinion, one of the most interesting drug classes is this drug class antisense oligonucleotides. These are currently being investigated in Alzheimer's disease. So ASOs, antisense oligonucleotides, They are synthetic nucleotides, so they are related. They're molecules that look like RNA and DNA. And if you want them to have an effect on the brain, they do have to be intrathecally injected. So that is one limitation. They have to be given through regular lumbar punctures. But when they enter into the cerebral spinal fluid, they can enter into brain cells, and they can specifically hybridize with a very specific RNA target. And ASOs can do a variety of things depending on how they're designed, but one of the things that they can do is they could specifically suppress the expression of an RNA target. So there are now ASOs that have been designed that can actually suppress the production of tau. So in theory, you have a way of teaching brain cells to make less tau. And a drug like this, BIB080, was just recently investigated. It's actually in a current trial in Alzheimer's disease, and it was just recently investigated in a phase 1 trial in mild Alzheimer's disease. Again, a disorder that is associated with its own kind of tau pathology. Patients with Alzheimer's disease tend to have very high tau in their cerebral spinal fluid. And in this phase 1 trial, this drug, this antisense oligonucleotide, really did seem to robustly decrease the expression of tau. So the CSF tau that was detected was suppressed by about 60%. And so this has led to a lot of discussion in my community about the possibility of using drugs like this within frontotemporal lobar degeneration with tau pathology. And I want to mention that a drug with a very similar mechanism is already in an ongoing clinical trial in patients with progressive supranuclear palsy. So as I mentioned, part of the reason why we haven't seen as many MAPT mutation carriers in clinical trials has to do in part with the rarity of the disorder. And this was a figure from a paper from 2019, so it's outdated at this point, but it's just to drive home the point. So it's reflecting the relative amount of mutation carriers with C9 progranulin and MAPT mutations really across the globe at major sites of FTD research. And the take-home message is that if you really restricted yourself to any one geographic region, it would be hard it would be really hard to conduct more than one clinical trial at any one time. You really need a large international effort to facilitate a robust clinical trial program in order to have a successful clinical trial landscape in MAPT mutation carriers. And fortunately, that's happening right now. There's active discussions about international MAPT mutation carrier trials being facilitated by this organization I mentioned earlier, the FPI. And in addition to needing that large international collaboration, you really probably need to rethink the way trials are conducted in general when you think about conducting a lot of clinical trials in a rare disease. To this point, we could probably take cues from the ALS community in how they've restructured some of their trials. This is a schematic from the Healy ALS platform trial that's currently underway. A platform trial is a special kind of trial in which one cohort of individuals can be enrolled under one protocol that can test multiple different regimens of therapy in parallel. So instead of having multiple unrelated clinical trials for multiple drug candidates, there's one clinical trial and multiple regimens and separate regimens tested within the same trial. And this is a much more efficient way to run a clinical trial in a rare disease. And the beauty of it is that each of these separate cohorts will share a placebo cohort. And so this is efficient in multiple ways. So you can theoretically test multiple, let's say in comparison to a traditional program with multiple clinical trials that are being run separately or staggered, you could test a set of drugs with fewer patients over a shorter period of time. And crucially, you could test this amount of drugs with fewer patients being assigned a placebo. So in a platform design, more patients are coming in contact with the drug candidate that may help to slow their disease process, which I think is really important in our patients. Another thing I want to mention, when thinking about how to plan out, the next wave of clinical trials in familial forms of frontotemporal dementia is, this is a really important point, that we now have all of this really important longitudinal data that we can leverage from these multi-site consortia that I've mentioned earlier, ALL-FTD and GenFI. This is an analysis that was led by my colleague Adam Staffaroni, merging together efforts from ALL-FTD and GenFI. And what Adam did in this paper was really look at all of the clinical data points that were available across ALL-FTD and GenFI, and put them together in one large statistical model to try to model out where each individual was along this axis of a hypothesized disease age, with 0 being set at the time of onset, or being conceptualized as the time of onset for symptoms. And modeling all of the data across, all this rich data across the consortia has a variety of potential benefits for clinical trials. One of them is that we can start to kind of drill in on how different mutation carrier cohorts might change over their lifespan, so that we could really push the envelope in planning clinical trials that are really trying to enroll patients who either don't have symptoms yet or barely have symptoms. And because we can have a more in-depth understanding of how clinical measures change over time, particularly in that interplay between symptom onset, between asymptomatic and symptom onset. And we can even leverage these models to think about how different biomarkers change. So some of these individuals that we're trying to enroll in trials and help, we may not have a good clinical measure to prove that a drug is working, but some of these models can help us analyze different biomarkers as proxy measures for disease efficacy, including imaging measures or fluid biomarkers. One potential fluid biomarker that might be used for this purpose is something called neurofilament light chain. Neurofilament light chain is a non-specific biomarker of neuronal injury, but it's really elevated in frontotemporal dementia. And we know that it becomes elevated as people are becoming symptomatic. So it tends not to be elevated in asymptomatic individuals. It's a little bit elevated in early symptomatic individuals, and it's very elevated once people become symptomatic. And for this reason, it's one of many candidate biomarkers for impending symptom onset. And using data like the data that exists in the GenFI cohort and in the ALL-FTD cohort, we could think about modeling biomarkers like neurofilament light chain longitudinally inside of these large sophisticated models, and really drill down on ways to use something like neurofilament light chain to plan out a trial in pre-symptomatic individuals or early symptomatic individuals. At least that's an idea. And what I'm talking about, in the last few slides was starting to get a little bit abstract. I just want to kind of hone it in with something a little bit more concrete. So there was a drug called tofersen that was just recently approved in ALS due to SOD1 mutation, so separate genetic cause of ALS, unrelated to C9 open reading frame 72. But the interesting thing is that in the approval for that drug, the FDA reviewed a variety of data and really looked at the drug's effect on neurofilament light chain on patients at the early stages of this disorder. And they really, in the approval, the FDA rationalized NFL as a reasonable predictor of clinical benefit from this drug, tofersen. And based on that approval, that was really cause for a lot of discussion in my community about the potential future of biomarkers like neurofilament light chain and other biomarkers that could conceivably use to support accelerated approval pathways in drugs within the rare disease space. I also want to mention another really interesting FDA approval for a drug called Scalaris. This was just recently approved for a totally separate neurodegenerative disease called Friedreich's ataxia. That drug approval, the FDA approved that drug for use based in part on the typical mechanism of a randomized placebo-controlled trial, but in support of the data from that randomized placebo-controlled trial, the FDA also reviewed open label data in comparison to data from historical controls. And so the fact that the FDA did that really, that was yet another thing that caused a lot of discussion in my community about what the future of of FDA drug approvals might look like for that very first disease-modifying therapy approved in frontotemporal dementia. It is possible that some of these sophisticated statistical models that I showed you earlier might be leveraged to help with future drug approvals. And maybe this is what the cure is going to look like in FTD. Maybe we will have a clinical trial in enrolling individuals who don't have symptoms yet. Most clinical trials to date have enrolled individuals who are already symptomatic, but maybe in the future we'll have clinical trials that enroll people that are pre-symptomatic or very, very early in their symptoms. And part of the efficacy signal of that drug may be more sophisticated than just a pure clinical efficacy signal. It may have something to do with pushing back what their hypothesized disease age is based on this sophisticating modeling. Anyways, that's the general idea. So with that positive note, I really want to thank you all for all of your attention, and I'm happy to answer questions about everything I talked about. Thanks a lot.
Thank you, Dr. Ljubenkov. We're going to bring Ms. Jenny back as well so that both can participate in a brief Q&A. So we're going to take some questions from the audience. If you haven't submitted a question yet, please use the questions box, and we're going to answer as many questions as time permits. We might not be able to get to all of them today. Dr. Ljubenkov, I might just start with what you were talking about at the end of your presentation there. We've gotten a few questions about genetic carriers participating in clinical trials who don't have symptoms. And I know you mentioned there was one trial that was recruiting people that the biomarkers were indicating they may be starting to or getting close to manifesting symptoms. Could you just talk a little bit about why more trials aren't accepting people who are not yet symptomatic, since they may be more realistically able to participate in a clinical trial?
Yeah, that's a great question. And I mean, there's multiple reasons. Some of the reasons have to do with potential measures of efficacy. If somebody doesn't have symptoms yet, it's really hard to know if a drug is having a clinical benefit. And so you really need to have a very good understanding of what the appropriate predictors are of impending disease onset in order to conduct a clinical trial in asymptomatic individuals. Or you have to really have done a lot of hard work in approving a good, reasonable proxy biomarker for drug efficacy. And this is a huge goal of organizations like ALL-FTD, GenFI, FPI, the larger landscape of this collaborative effort is to really figure out how to prevent disease. I mean, we do want to run clinical trials in symptomatic individuals too, but we also want to find a way to prevent disease in individuals who carry risk factors. And it's just been difficult to devise those trials to date.
Understood. We've also gotten a few questions about costs that a family or an individual might incur either by participating in a clinical trial or through genetic counseling and testing. So I was hoping we could answer those both. Maybe Kim, if you wanted to start with the costs that might be associated with testing and counseling, and if there's any ways that people can receive that for free, or if there's ways to get that covered.
Sure. So if you're working through a research protocol, in theory, there should be no cost to the participants. But if you're interested in pursuing genetic counseling and genetic testing and limiting the cost to yourself, there are a couple of different ways to access testing. There are some sponsored genetic testing programs, which typically are paid for by a biopharma company or sometimes an advocacy organization. The reason they're paying for the testing is they want to have more people available for research. If it's a biopharma company that's trying to develop a drug, certainly knowing how many people are out there who could benefit from the drug would be good for the company to know, as well as knowing how many people might be available to participate in research or be interested in participating in research. So there are these sponsored testing programs. Most of the sponsored testing programs will offer complimentary pre and post-test genetic counseling. So that's something I didn't really touch on when I was talking, but it's important to have genetic counseling before testing, because genetic test results are often not straightforward, and there are certain sort of financial, legal, social aspects that need to be dealt with prior to pursuing genetic testing. And a genetic counselor can help families navigate all of that. After genetic testing, it's also highly recommended to have genetic counseling because you need somebody to help you interpret those results and help you plan out the next steps. So sponsored testing programs are one opportunity to have no-cost testing. And then, of course, research protocols. And a lot of the research protocols do include genetic counseling, like the ALL-FTD study. They do have genetic counselors available at most of those sites. Okay.
Great.
There are
Thank you.
also labs. One last thing, sorry to interrupt you. There are some labs that will offer reduced cost testing for families that choose to pay out of pocket. So they try to limit the expense. If a family is going to be paying cash for their testing, they want to make sure that they aren't causing a burden for the family. Okay, sorry to interrupt.
Yeah.
No problem. Thank you. And Dr. Ljubenkov, I wonder if you could answer the similar question about being a clinical trial participant.
Right. So participation in a clinical trial should be free. That is generally the case. There are circumstances in which, unfortunately, there are programs that are listed as trials on clinicaltrials.gov, and they are essentially pay-for-play models. I have problems with the ethics of that, and all of the clinical trials that I mentioned, you do not get charged for participation in that trial. Now, there is still burden that is incurred by being part of a trial. You have to travel to the site. Oftentimes your caregiver has to take time off of work. There are still other costs that are incurred just by being part of a trial. And most trials try to set aside a budget to mitigate that, to recoup certain travel expenses and give patients stipends to try to handle that. Especially sometimes if it's an investigator-initiated trial, and it's not privately funded, there may be a limited budget to do that, but we do also try to recoup that that burden as well. But really, if you're part of a clinical trial in frontotemporal dementia, you really should not be paying to be participating in that trial.
Thank you. And finally, I wonder if you could talk a little bit, Dr. Ljubenkov, you mentioned a number of trials, for example, for people with FTD caused by the progranulin gene. Do you have any tips on how people should choose a trial if there are a number of trials available to them for their specific, either their specific gene or for their specific diagnosis?
Gosh, that's a really tough question to end with. I mean, I think that it really depends on the person's priorities, in terms of like, all the different trials have different levels of how invasive they might be, whether they be gene therapy or infusions of the drug regularly. So, either get a suboccipital tap, early on, which is a relatively invasive process, or come back for multiple infusions over a prolonged period of time. I think it really is something that a person needs to talk about with their neurologist. And I think the model that's worked best is a lot of individuals who are enrolled in these clinical trials are already known to researchers within the ALL-FTD network or within a consortium within their geographic region. And those consortia, in addition to conducting clinical research, they are an avenue to access individuals who have expertise in genetic forms of frontotemporal dementia. And oftentimes, I see a lot of patients for ALL-FTD here at UCSF, and oftentimes that's a large part of what I do here during assessments is walking patients through the different trial options that are available and helping. There's no right answer for everybody. No answer that's right for every single person. Everybody has a different thing that's right for them based on what they prioritize. And it really takes getting to know a patient and having some expertise in the field to counsel them through that.
Thank you. And I know there are a number of questions we weren't able to answer live, but we do encourage you to reach out to AFTD or have your patients reach out to AFTD. And I really want to thank you both, Dr. Ljubenkov and Kim, for sharing such valuable information with us. Before we wrap up, I have a few updates from AFTD. We have recently launched a support group for asymptomatic people who have tested positive for an FTD gene. This group offers community, connection, and a safe place for members to share their experiences living with and managing their feelings around their genetic status. So if you have patients living with a genetic risk for FTD, or if this impacts you and your family, we encourage everyone to contact the AFTD helpline to get more information about this group. And there are a number of ways for people diagnosed with FTD to participate in research, as you've heard today. Many FTD clinical trials are underway and seeking participants. Studies that include experimental treatments to target FTD symptoms, as well as potential disease-modifying treatments for people with sporadic and familial FTD. More information about studies seeking participants is available at the AFTD website, and people living with FTD or those who have a connection to FTD can get updates about relevant studies emailed to them by signing up for the FTD Disorders Registry. So you can visit ftdregistry.org to learn more about getting those updates and even participating in remote research. In addition, AFTD recently launched its new Advocacy Action Center. We encourage you to visit the URL on your screen and sign up to be kept informed of ways to take action at both the federal and state levels. You can also get involved in advocacy by sharing your personal story. Our next webinar will be held January 18th and will focus more on gene therapy and FTD. This webinar will be a joint presentation with the FTD Disorders Registry and will be intended for the lay audience. Registration will open early in the new year. While CME credits won't be available for this particular webinar, we will be resuming our healthcare professional education webinar series in 2024, so stay tuned for details. And again, we want to thank you for joining us, and thanks again to Dr. Ljubenkov for sharing his expertise, and to Kim. And thanks also to Rush University for allowing us to offer continuing education credits for this webinar. If you have additional questions or comments about today's presentation, please reach out to us. We would love to hear from you. Thank you all.